Okay, let's talk about the first kingdom, which is kingdom prokaryotes. So this is a cell of a prokaryote. All prokaryotes contain plasmid, cell wall, plasma membrane, cytosol, ribosome, as well as DNA strand in nucleus, which is a region that contains DNA, region in the cytoplasm. Okay, and certain prokaryotes contain flagellum for locomotion to help the bacteria to move, and certain prokaryotes also contain capsule to act as a protective layer to the bacteria or prokaryotes, and certain prokaryotes or bacteria also contain fimbriae, which help to attach the bacteria to attach to the host cell. Okay, prokaryotes are divided into two domains based on the structure of the cell wall. The prokaryotes that has peptidoglycan is classified under domain bacteria, whereas the prokaryotes that has no peptidoglycan is classified under domain Archae. An example of domain bacteria is E. coli. An example of domain Archae is Sulfolobus species. These are the characteristics of prokaryotes. First, prokaryotes have no nucleus. So it means that the DNA in prokaryotes is located within the cytoplasm in a region that we call as nucleus. So nucleus is a region in the cytoplasm that contains DNA. And other than that, prokaryotes also have no membrane-bounded organelles. So what are the membrane-bounded organelles? Membrane-bounded organelles are lysosome, endoplasmic reticulum, Golgi apparatus, chloroplast, nucleus, and mitochondrion. So prokaryotes do not have these organelles. Other than that, prokaryotes are also unicellular. Okay, as other characteristic of prokaryotes has circular DNA that is called as plasmid, and DNA in the bacteria or prokaryotes is not associated with histone protein. So histone proteins are proteins that in which DNA are coiled around it. So all bacteria does not has histone protein within their DNA except for Archaea. And other than that, prokaryotes also have smaller ribosomes compared to eukaryotes, which is the 70S. So S is a unit that we use to measure the size of protein. And cell wall of prokaryotes composed of peptidoglycan, as opposed to cell wall in plants. Cell wall in plants consists of cellulose, whereas the cell wall in fungi is composed of chitin. And flagella is also simple in prokaryotes. So prokaryotes flagella consists of this arrangement we call flagella in eukaryotes consists of arrangement that is called as 9+2 microtubule arrangement. So prokaryotes lack of this structure in their flagella. So the flagella of prokaryotes are simpler than this. Other than that, prokaryotes also reproduce by binary fission, which is a type of asexual reproduction. So what is the difference between binary fission and mitosis? In binary fission, it does not involve the division of nucleus, because as you know, prokaryotes do not have nucleus. So it means that they just consist of cytokinesis, which is the separation of cytoplasm. So it means that in their division, there's no such thing as metaphase, anaphase, prophase, and so on. Okay, let's compare the two domain, which is domain bacteria and archaea. Okay, let's talk about cell. Let's look at the cell wall structure. In bacteria, the cell wall consists of peptidoglycan, whereas in archaea, there's no peptidoglycan in the just cell wall. It means that archaea has cell wall, but the cell wall is not consists of peptidoglycan. And then after that, let's take a look at histone protein. In bacteria, their DNA is not associated with histone protein, whereas in archaea, the DNA is associated with histone protein. Let's move to diversity of bacteria. We classify bacteria based on the shapes of the bacteria. Okay, the first one is spherical or coccus. So from the name you can see that the shape of the cell is spherical. And other than that, some bacteria shape are rod or bacillus shape. And certain bacteria are spiral or helical shape. And certain bacteria are vibrio, means that the shape is more like comma shape. Okay, and then lastly, let's talk about the importance of bacteria. So the function of bacteria or the importance of bacteria is to recycle chemical elements in ecosystem. For example, rhizobium is fixed nitrogen. It means that rhizobium will take the nitrogen in the environment and convert it into organic form. For example, like nitrite or nitrate, which will be used by the plants, and the plants will convert those those compound to form protein. And other than that, bacteria also act as a decomposers. It means that it recycle the dead organic materials. I mean, if there is any corpse, any animals or plant, any dead animals or plants, the bacteria will sort of cut, can call it as cut it into pieces, so smaller pieces. It will turn it into simpler form. And then bacteria also release nitrogen and carbon gases back into atmosphere. And normally carbon is released in the form of carbon dioxide, whereas nitrogen is released as a nitrogen or N2 gas. Okay, other than that, bacteria also have important role in symbiosis, especially in our intestine. So bacteria that are located in intestine are called as enterobacteria. So the function of enterobacteria is it helps prevent the growth of harmful bacteria. And other than that, it also helps herbivore to digest cellulose. For example, cow. Okay. So in the stomach or in the digestive system of a cow, actually it has the bacteria. So the bacteria that will convert the cellulose that is consumed by cows to simpler form that can be absorbed by the cow. So as humans don't have the cellulose, don't have the bacteria that can convert the cellulose into simpler form. That's the reason why when we eat vegetables or any form of fiber, we cannot digest it. It will come out through our feces in the form of in the same form. And then the next importance of bacteria is as pathogen. Certain bacteria, as you know, can cause disease. For example, salmonella, which can contaminate food, can cause typhoid fever. So the main symptoms of typhoid fever are vomiting, and then rash, and diarrhea as well as stomach pain. And the last one, bacteria are also used in research and technology. For example, as you learned in previous semester, we use bacteria in recombinant DNA technology to carry human insulin gene so that the bacteria will produce insulin for us. So that's the example of importance of bacteria in research and technology.